Je vais faire plein de franglais, hein, je suis désolé, mais c'est pas grave. Des bile ducts in vitro.
Oh, pas de
Et,
problème.
et évidemment, le travail depuis 4 ans de Harini, Rajendiran, donc au Mechanobiology Institute, et de moi-même. Et je viens donc de bouger au Turing Center à Marseille. Voilà. Donc, ce qu'on a essayé de faire dans ce travail, c'est de reconstituer in vitro la partie portale d'un lobule en essayant de créer des bile ducts qui s'allongent et en recréant un maximum de la diversité cellulaire qu'on peut retrouver dans ce portal area. Et donc, pour ceux qui pourraient encore l'ignorer, la particularité de portal area comme ça, c'est que vous avez le canal biliaire, pardon, la veine portale ici, et que le long de la veine portale, vous avez le bile duct qui est... I'll switch to English, sorry, I cannot do it in French, I haven't done it for a long time. So you have bile duct lining the portal vein here, and you have the biliary artery also lining on the other hand. The nice thing about the bile duct is that it follows the portal vein wherever they go, and so you have the portal vein that sort of scaffold the growth of the bile duct in the adult liver. And this paper by Takashi in 2015, they even show that when you have morphological defects in the vein, in the portal vein, then the bile duct would follow exactly the portal vein structures. So one of the reasons for that has been looked in details, and including by Frédéric Lemaigre a lot. It comes from the fact that you have along the portal vein, you have, during the development of the liver, you have these mesenchyme layers that secretes a series of growth factors, TGF-beta 1, 2, 3, activin A, and JAK1, that signals to the surrounding hepatoblast, and the surrounding hepatoblast will differentiate into into cholangiocytes, okay? And so that's what you see, the different steps that are, Frédéric, for example, has reported and looked in details, is that you have the portal vein here, you have the secretion first of the paracrine, signaling from the mesenchyme around the portal vein that leads to a differentiation of hepatoblast into cholangiocytes. And as time goes on, these cholangiocytes, they form a ductal plate, which is a scattered monolayer of cholangiocytes. And then they start forming these tiny lumens. And the lumens eventually grow and anastomose into longer tubes to eventually collapse into one large tube that follows the portal vein. So this is what is reported extensively in mouse. It's been, as far as I understood and could find, relatively less looked at into humans for obvious reasons of how you can follow this sort of processes in vivo. So of course, as you know, one of the aspects is to reconstitute the liver development and liver structures in vitro, and especially when it comes to bile duct. And so there are different ways of doing so, starting from very simple 2D culture, where you start with hepatoblast from either that you collected directly or that you derived from definitive endoderm, and then you can culture them in 2D, and then you get a beautiful epithelial monolayer, but then, of course, they don't form tube. Now, if you take these cholangiocytes, when they are differentiated and you place them into matrigel or any sort of other kind of 3D matrix, they form these beautiful cysts, which are monolayers of epithelial cholangiocytes, forming this big balloon. And the reason why they form big balloon is because they secrete salt, and the salt creates an osmotic pressure inside the monolayer, and then the monolayer expands isotropically everywhere in the gel, and then you get this round shape monolayered cholangiocyte organoids. So this is how we have made the first cholangiocyte organoid. Now, If you want to achieve tubes, you have another way to do it. It is to actually create the tube yourself. So you can use microfabrication. And for example, in this system, you have two channels connected, two cavities connected by a channel. The channel is the tube itself. And within the tube, you grow the cholangiocytes. So they're happy. They coat the surface of the tube. They're polarized. But of course, the morphogenesis process of that system is very limited, specifically because you impose the geometry into which the cholangiocytes grow, and you impose the tube formation. Now, there's another way that you can play around with biochemical cues and biophysical cues. It's actually to pattern the extracellular matrix in 2D. So depending on how you do it, you can end up, if you differentiate the hepatoblast, you can end up with variation in differentiation patterns along this planar extracellular matrix deposition, or as in the team of Pascal Dupuy-Williams has done, they pattern this branched architecture of trees, and then they grow the primary cholangiocytes on them that eventually fold and create tubes that are branched following the ECM pattern. Now, very recently, there's been another kind of organoid developed, which grows with having this sort of cyst-like structures and place them in a medium where you change somehow the growth factors and place them in extracellular matrix. And now you have this beautiful branch structure that grow and branch with a budding tip here that grows over and over inside the matrix. So these things reach sizes of few millimeters. However, they're not scaffolded, and the branching pattern here is very nice, but it's not really like what happens in the liver either, because it's very similar to what happens in any epithelial cells, and especially in breast cells, where you have a growth from the tip and branching events. So all this shows that cholangiocytes can actually create bile duct, but the morphogenetic process by which they can reach the bile duct in vitro is not very equivalent to what happens in vivo. So we had this hypothesis that what was missing in all the system was precisely to have a guiding cues, which were provided by the portal vein. So the idea was to change and replace the portal vein and the surrounding mesenchyme by a rod made of gelatin metacrylates. So it's gelatin cross-linked with metacrylate groups. And on that rod, we grafted, chemically, we grafted the activin A or different growth factors. And the idea is that if you get such sort of rods, can that mimic the effect of the portal vein, and hence lead to the differentiation of the hepatoblast if the hepatoblasts are placed on these rods. So this is what we did. We created those rods in a relatively simple manner. You take a hollow tube, you fill this long hollow tube with gelma, you cross-link, then you chop by hand tiny bits of the hollow tube. So typically the hollow tube is 400 to 500 micron in diameter. And when you chop them into bits, you reach sizes between 1 and 1.5 millimeter, depending as you want. And then you expel the center part of the gelatin inside, and you functionalized with heparin, biotinylated heparin, sorry. And then you graft the growth factors after that on them. So now you can play with different concentration of growth factors and different kinds of growth factor, cocktails of growth factors that you put on the gel. So we'll deal with three kinds in the whole process of the talk. One where there are no growth factors, just the gelma rod itself. The other one is activin A and FGF10. And the third conditions will be activin A, FGF10, and JAG1. Once you have these rods, what you do is you start with HIPSC colonies that you push to hepatoblast through the typical 12-day process. And once you have the hepatoblast, you seed them as single cell onto the rods. And then what happens after 1 day is that the rods are getting covered with a relatively thick layer of these hepatoblast. And if you wait until day 8, this is what you get, a time lapse. You start having this bulging structure that emerge from the rod and that start pulsating. So this is what is alike bile duct, as I will show you. So what we did then, we decided to look at whether these processes by which the cells differentiate mimics what happens in vivo. And the first thing was to fix the cells at day 1 after seeding. And this is what you get. You get with fluorescence imaging, you get these rods here in the center. And then in green, you have this monolayer of cholangiocyte cells. So here it's a label for cytokeratin 7, but we checked for other markers. And so you have this monolayer of cholangiocytes lining the rod, and then you have these other HNF-alpha positive cells, which are like the hepatoblast. And they are SOX9 positive, they are dappled here. Okay, so that's really mimicking the sort of initial stage where you're creating a ductal plate around that rod. Now, we could vary and change the density of the differentiated scattered cholangiocytes around the rod by changing the density of ligands and the kinds of ligands and growth factors that we put on the rod. So to our surprise, if you put the hepatoblast directly on the rod with no ligand, no growth factors, you still get some local differentiation of hepatoblast into cholangiocytes. And this is because our hepatoblasts are already prone towards cholangiocytes lineages. So even if you don't do anything, they don't stay as cholangiocytes, but they have spontaneous differentiation. Now, on the other hand, if now you add activin A and FGF10 on the surface of the rod, you get a much more denser monolayer of cholangiocytes lining the rod. And if you add now JAG1, you get something way more structured and even more covered. So all this is somehow quantified here, where you look at the density of ligand and you look at how much area is covered, how much of the rods are covered, how much area of the rods are covered. Yes. Now, this is for day 1. So you get to the stage where you have this monolayer of cholangiocytes around the rod. Now, if you wait for longer time, now I'm zooming in on specific events along the rods. You start seeing at day 3, you start seeing this sort of almost closed lumen, which are multi-layer, well, you can call it multi-layer aggregate of cholangiocytes, but you still have an opening in the center. At day 4, you see that these openings are getting bigger and bigger. At day 5, you have these lumens that start filling each other and finding each other and accumulating. And now at day 6, you have this very large, pronounced lumen that grow, and it's pretty much what you have seen pulsating on the rod when I show you this in a minute ago. So this also mimics, as I already described, what Frédéric has reported as a process of how you get the hepatoblast, differentiating into cholangiocytes and going all the way to the rod. The only difference that we see from the in vivo reported case is we never see these heterotypic lumens where we have on one side the cholangiocytes, on the other the hepatoblast. In all the cases we have only lumens constituted of cholangiocytes only. Now we tested for several other markers to look at what these lumens are made of, and they are really made of. They have the right staining for cholangiocytes all along after day 6. But the most surprising and the most, I think, beautiful thing that we were not expecting is precisely this deposition of laminin laminin basal layer, extracellular matrix layer, at the edge surrounding the lumen. So what you have here is the laminin in purple, and then you have cytokeratin 7 in green, and then you have Z01, not cytokeratin 7, Z01, and you see that the cholangiocytes are underneath the basal layer, and the rest are other cells which are very well polarized at the apex of the lumen, of the cell sheet. So just to put this in perspective, there is no extracellular matrix added to the cell culture. So there is no matrigel. The cells, they're only filling the extracellular matrix, either that they secrete themselves or the extracellular matrix, the gelatin that is on the rod. But we do not embed the entire system into any extracellular matrix. So you get this lumen and multi-layer structure here, which is well polarized at the very top. Now, if you treat the rods with different growth factors, you have a very different morphologies in the end for the lumens and the equivalent of bile duct that you get. So if you have no ligand at all, you get almost exclusively this nice, if you wish, but big spherical lumens that are either lining the rod, but most of the time they accumulate into this sort of big lump of cells, and they never really make tubes. They really make these spherical things that never really merge. Now, if you have activin A and FGF10, you get what I showed you before, where you have these elongated lumens, which are usually not, I'll come back to that, but they usually, you still see different lumens merged together, but they are still kind of elongated. And now if you use activin A, FGF10, and you add JAG1, in this case, you get this very long lumen spanning half of the rod, sometimes even the totality of the length of the rod. Surprisingly, in the end, after 7 days, in this case, you almost get only one big lumen only, and the rest are small ones scattered, but there is one big lumen and one big duct going around the rod. So these are the different markers we tested. We tested other ones, but just to show you that these guys are really made, the lumens are really made out of cholangiocytes. Now we faced some kind of problem. And the first problem we had was that to image the rod, as I told you, this is about 1 millimeter long, 1.5 millimeter long, and 400 micron in diameter. And to do the confocal imaging of that, at best, in the best case scenario, it took us 4 hours per rod to be imaged. And then you get a lot of optical problems because you have to image far into the sample, and the quality is not always great. So one idea that we had in collaboration with Matt and Byrne was, and in the Brit lab in Brisbane, sorry, it's not in Brisbane, it's Perth, was to use OCT. So OCT is optical coherence tomography. It's the typical imaging technique that you use to look at retina. And what it does in hospitals, but nowadays there are ways where you can look organoids with this OCT system. And what it does, it's a sort of interferometer imaging. And then what it gives you the contrast of the optical index in 3D of your sample. So you can do it live. It's label free. You don't have to stain. You can image the cells live, of course, and it's very fast. It takes only 20 seconds to image the entire rod here. And because of that, we can image 96 well plates that you see here. Each of the wells here have one rod and one organoid. And so that was, for 20 seconds per well, it was relatively easy to get the 3D images of these rods out once you know how it works. And so once we had that, we devised a way to extract the stacks from the interference patterns to get the image that is relatively stereotypical for this kind of imaging. But after that, we had a whole process of realigning all the rods and all the stacks together and training different kinds of AI-based segmentation processes so that the system discriminates between the rod area, the cell area, the background area, as well as the luminal area. And all that, in the end, we were able now to reconstruct in 3D for each of the organoids and each of the rod, the luminal area. So what you see in this film are each of the rod, I think, is very obvious, but the other pieces that you see here in color are the actual bile duct lumen, and each is color-coded for its identity. So each color corresponds to a different lumen, as identified by the software. So once we have that, now it becomes easier to look at precisely the dynamics of how these lumens are actually formed and grow. So again, if we go back at day 1, we get this multitude of tiny lumens formed in it's day 2, actually, not day 1. So you get between day 2 and day 4. So what you get is these tiny lumens that we can now quantify. So typically we quantify 2 things for them. We quantify their sphericity, so how round they are. High sphericity means that the lumens are very round. Low sphericity means that the lumens are elongated. We quantified their longest axis, how long they are in the longest direction. And then we also checked how far away they were from the rod. And so if you look at day 4, you have all these lumens from different batches and different rods. And you see that most of them are relatively round with a high sphericity. And we define 2 regions, one which is proximal to the rod, which means that the lumen center is less than 30 micron away from the edge of the rod. So 30 micron is approximately 2 to 3 cell layer. And you see that you have quite a number of lumens that form in the very vicinity of the rod. Some others are further apart inside the big chunk of hepatoblasts that are there. Now, if you wait for a longer time, what you get is actually pretty much the same number of, for longer time means at day 5, you get same number of lumens. But as you start seeing, this is day 4 and this is day 5, you see that the sphericity goes down for those close to the membrane, to the rod, sorry. And that means that now they grow and then they start to elongate because they are less spherical and their length is also increasing. And this is what you see in this image, where you see that now these lumens have grown. And you still have plenty of the other lumens aside from that proximal region. Now, if you wait even longer, at day 6 and onwards, what you see is a complete change in the morphology of the lumens. You see very fewer lumens on the rods, but now they are very elongated, very long, very elongated. Of course, a few remain round and far away from the rod, but those who are very close to the rod, they really are much, much longer and very elongated. So that we can quantify and follow in details. And what we found was that these lumens always, in 80% of the cases, they align with the major axis of the rod. So you never have lumen coming across. You have them really going along the rod almost all the time. And in the end, if you wait longer, in many cases, you get almost one long exclusive lumen. The other ones are scattered. So when we looked more in details how this happens, this is live imaging. Well, you have only one fixed time point, but this is the cells are alive, and this is a Z-cut, and you see that these lumens are actually convolved, right? They create these long tubes, but the tubes are merged cavities all along the area. And you have, typically, this is a zoom where you have one lumen here that connects to another one through and this is a bunch of cells. Most of them is a monolayer of cholangiocytes, and the rest is some other cells. And so now you have this anastomosis of the spherical lumens that connect to each other through these tiny connection zones. So when we were there, we were super happy to be able to look at, precisely the live imaging and quantify those lumens and see that the bile duct was growing. Somehow I wanted to make one specific comment on the morphology here, which I'd like to come back in the end. But you see that we are somehow obtaining, depending on the type of ligand that we put and the type of growth factors, we're obtaining lumens that are almost like a real tube, all the way to lumens that to bile duct that look almost like polycystic, where you have this inclusion lumens everywhere, and where the longer lumens, at least for a fixed time point, in this case, they do not really merge together. They're rather having these distinct identities rather than creating a long tube. So this is somehow controlled, at least to some extent, by the amount and the characteristic growth factors that you put on the rod. Now, we're happy to be able to reconstitute this. We're happy to see that it follows somehow the path that it is reported to follow in vitro, despite the fact that it's for human cells. Now, our surprise, or what was not expected at the start of the project, was when we did single cell sequencing at day 8 of these lumens, of these organoids, was single cell sequencing was showing that not only did we have cholangiocytes, which are these, where are the cholangiocytes? I missed the, oh yeah, here is the labeling. Okay, so the cholangiocytes are actually these purple or pinkish cells here, but we also had hepatocytes. We also had liver progenitor cells. We had, of course, hepatoblast, because we started with the hepatoblast. But at the same time, we had also endothelial cells, and we had hepatic stellate cells and some unknown clusters. And so when we tried and look at the different staining, and we stained, for example, for albumin, and we could see that within the bulk of cells that surround the actual bile duct-like lumens, we had cells that started expressing the albumin protein, which then we'll still have to confirm that, but are likely to be the hepatocytes. Now, when we looked at also in other kinds of markers, I think here we are looking at V-cadherin and cytokeratin 7. So cytokeratin 7 stains for these bile duct-like structure. Here they really look like polycystic. But you see that now you have two very characteristic things that happens every time. Along the rod, between the rod, which mimics the portal vein, and the lumen here, which is the bile duct, you have this monolayer of cells that express V-cadherin and that really look like endothelial cells. So they sort of intercalate between the rod and the bile duct. And now, away from the bile duct, on the other side of the bile duct, you have these lumens here that was where, you see, if I zoom in, you have lumens. And these lumens are also positive for V-cadherin. And so they also are lining with endothelial cells. So we think that they represent either veins, kind of veins, or kind of arteries. We don't know yet which sort of lumens they are exactly. And so these lumens that you see away from the rod and are relatively big, these lumens that appeared in our characterization when we used the OCT. So what we initially thought were sort of failed bile duct just happened to be actually lined with endothelial cells everywhere. So we think that now we have all these different stellate cells, endothelial cell, cholangiocytes, hepatoblast, hepatocytes, and liver progenitor within these organoids, which makes it really, I mean, as close as we so far can do to the portal area of the liver lobules. So we think that this sort of system can be a first step in trying to reconstitute more of the liver lobule. I really insist that to obtain this, there was no added culture medium, there was no added extracellular matrix. It's pure self-organization coming from grafted growth factors on the surface of the rod and seeding of hepatoblast on that surface. The rest is just a maintaining medium where cells would grow, migrate. So now I would like to come to the conclusion and some remarks. One of the things is I think this system really shows that you can mimic the paracrine signaling coming from the portal vein in order to create these bile duct-like and multicellular-like organoids of bile duct. And I insist again that it's very important that the growth factors are grafted on the rod and not released. So there's no gradient. It's really, really done by paracrine signaling. Okay, probably it looks like, you know, if you're not looking too close to it, but it really looks like the portal triad that we're starting being able to build. The important point also to remember here is, I think not only do we mimic in the end the sort of morphologies of the portal triad, but we seem to mimic also the developmental process by which you reach these different structures. So that's the important point, where we are really trying to know the developmental paths and how the different growth factors actually influence each step of this developmental path that we can recreate in vitro. We can image all these systems and quantify them in HCS format due to the OCT system, which is really nice. And I think that's something to keep in mind as a technique to image any sort of luminal structure inside organoid. And pretty much there are luminal structure for every epithelial organoid, including intestine, gastric, pancreatic, and so on. So that's very, very powerful technique that is, I think, underused for that purpose. And if you play with the different growth factors combination on the surface of the rod, then you end up with different morphologies and different morphologies of the bile duct. And in particular, some of them look polycystic when you have less JAG1, clearly. And so one of the expectations that, some of the expectations that I have by talking to you is, now we're in the process where we can really nicely mimic these systems, but we probably need some additional expertise to really dive more deeper into how morphological what are the morphological similarities between our cells that we develop or that self-organize in the case of this organoid with in vivo system, and in particular in human. This is really something that we were looking for help with, and I'd be happy to discuss with you. And at the moment, we have some ideas, but if you have some other ideas of what the applications of such a system can be, we are also looking for possible applications of what we can do with this parallelizable and quantifiable organoids. And with this, I thank you very much for your attention.